Good morning, and welcome to the BaseSpace Push-Button Bioinformatics Webinar Series. I'm Raymond Tikotsky, your webinar moderator, and today's topic will be assembling and analyzing Illumina data in DNAStar Lasergene Software. Our special guest presenter will be Matthew Kaiser. I'll turn it over to Matthew in just a moment. But first, I want to give you a little background on BaseSpace. So what is BaseSpace? It's a solution to seamlessly and securely upload, monitor, analyze, and share your genomic sequencing data. Its primary value or benefit to you is that fundamentally it makes genomic informatics analysis easier. Some of the stats about BaseSpace uploads and the fact that it's reached critical mass are over 40,000 sequence runs have been uploaded. We have over 6,000 registered users now. And 75% of all new MySeqs that have been connected are uploading to BaseSpace, and 50% of those are automatically sending their sequences up to BaseSpace every day. 10% of those now are actually sharing their data across institutions. And there have been nearly 5,000 app test drives so far. As you see, we've included many new apps, and we're adding more every day. On May 15th, we've included several new applications, plus we introduced iCredits. One iCredit is equivalent to $1, and this enables not only the free apps that we had before, but also now paid apps as well. So now I'd like to introduce Matthew Kaiser. Matthew Kaiser is a next generation sequencing application scientist for DNAStar, where he has spent the past 9 years helping scientists address their sequence assembly and analysis challenges. Matt supports customers in next-gen sequencing applications using DNAStar's broad software toolset, including templated and de novo assembly projects, transcriptomes, exomes, metagenomic assemblies, GWAS, RNA-seq, ChIP-seq, and similar workflows. Matthew?
Thank you, Raymond. So Raymond listed a lot of different workflows that our software can support with Illumina data. Today I'd like to show you a couple of those workflows. We'll start with an introduction with some PowerPoint slides, and then I'd like to move over to my desktop and we will look at some live software demonstration. So DNAStar has been a player in bioinformatics software for over 25 years. Our founder is Dr. Fred Blattner, who back in the 1990s first sequenced the E. coli genome. Some of the scientists that worked with Fred on this genomic project are still with DNAStar, so we have long-standing expertise in developing software for sequence assembly and analysis. We support Sanger next-generation sequencing platforms, including Illumina. Our software runs on Windows and Macintosh computers, on Linux computers, and also the cloud. And we'll also take a look at BaseSpace and our seqman engine towards the end of the presentation today. So DNAStar produces research-grade software, and that's represented well in the cited literature. Since 1985, we've been the most cited bioinformatics software, more than double the citations of our nearest competitor. So our software is powerful, it's made for researchers, and what I'd hope to show you today is that it's also very easy to use and accessible to regular biologists. So the power of the software is evident with some of the benchmarks that we have. And these are some benchmarks with Illumina sequence data. And of course, the first data set that we'd like to focus on is the human genome. That is the ultimate benchmark for next-gen sequence data. And we have an assembly of 1.5 billion reads, significant amount of coverage, 43x. And our assembly time is down now to about 13 hours. And that's really a tremendous performance. This is done on a computer that you could purchase at Dell for under $3,000. If you can build your own computers, we can help customers with that, and you can drop this price below $2,000. So really just a sturdy desktop computer. We can get excellent performance. This assembly time includes not just the alignment of the reads to the reference, but we also generate an output file in a BAM format, do all the statistical SNP analysis. So we have SNP files, annotation files, and so at the end of this 13 hours, you're really ready to begin your analysis. You don't have to run additional algorithms or additional reports that take additional hours. Smaller projects like human exomes may take a little over an hour each to complete. We can multiplex these projects into single assemblies. So we have our second benchmark, which is actually a project that we'll look at today. It's 8 human exomes aligned together to the human genome, very deep coverage, approaching 500x, and that's about a 10-hour assembly. Smaller projects, smaller genomes and E. coli genomes can be measured in minutes oftentimes. RNA-seq experiments, just a few hours. So the software really has tremendous capacity and speed, and that's something that can be very beneficial so that you can turn around and have a high throughput assembly and analysis, again, on very reasonable hardware. The software is also very flexible. It can accommodate a wide array of different workflows. There's actually a number of different algorithms that help support the different workflows. That includes genome resequencing, exome and targeted resequencing. And of course, with targeted resequencing, that could be a handful of genes, a few hundred exons. Oftentimes, they can be sequenced very deeply. So the algorithms have to handle hundreds or thousands of X coverage across areas of interest. RNA-seq data sets. We do an excellent job with de novo transcriptome assemblies. Folks working in non-model organisms oftentimes will sequence the RNA and want to cluster and find novel genes in some non-model organisms. So again, a great workflow here. We aren't going to look at that today, but I can give you more information on that later. There's also automated bacterial genome closure, and this is a novel workflow that DNAStar has developed recently, and it allows us to align our sequence data to the best possible reference genome, and then in an automated fashion identify the novel regions in the sequence strain and do a de novo synthesis in those novel regions. And of course, we can do this manually, or we can use DNAStar software and have an automated approach and algorithms that can do most of the work for you. We also have de novo genome assembly capability. Most of our customers focus on microbial genomes or smaller eukaryotic genomes. We have a very stringent, very accurate de novo genome assembler. So these are a number of different workflows. The ease of use I would like to show you by going into the software. So at this point, I'm going to jump out of the PowerPoint and share my desktop. And so I'm going to go here and launch seqman Engine. And so seqman Engine is, again, it's a 64-bit program. It runs on Mac, PC, and Linux. It also now is available on a DNAStar cloud as well as BaseSpace. So it's very accessible. There's a number of different ways to use seqman Engine. This program is really a project setup wizard. It allows us to set up projects of all different types. And so our usual starting point is to create a new assembly project. And this wizard then it guides us through the project setup. So the idea is that we don't have to be experts with how to run assembly algorithms. And really the hope is that a brand new user can load the software and get a very good assembly right from the start, right out of the box. We can choose our project type, and again, as we start to pick the different project types, that will set different optimal parameters. You can see we have a genome assembly or targeted resequencing or exome assembly, transcriptome, ShipSeq, metagenomics. We have a new workflow, viral host integration, that locates retrovirus integration sites in the host genome. And so again, depending on what you pick, it will start to set optimal default parameters for that pathway. And also the tree branches differently. I'll just show you If I select genome assembly, I actually have 4 different options for genome assembly: a templated assembly with a BAM output, a de novo assembly, our reference guided assembly with gap closure, and another specialized assembly option. If I pick targeted resequencing or exome, I have one choice, and that choice will be generate a BAM file output. BAM files are extremely efficient. We can open human chromosome 1 on our laptop and do our analysis, and so it is the ideal file type for variant analysis when we're aligning to a reference. At this point, we set up our project files. We can name our project, select an output folder, and so here we can pick where we want all our files to save to. The temporary file location is really an indication, a little bit of how the algorithm is going to work. It's going to use CPU, RAM, and disk space to process all of the data. We can go to the technical requirements here, and this will, I won't go into this in great detail, but for reference-guided assemblies, different project types, it's essentially the same core computer, 16 gigs of RAM, but where they differ is the amount of disk space that's needed for ever-growing data sets. So we can get some more information there. And these temporary files, again, it can be an internal drive, an external drive, doesn't matter. Drive space is economical these days, so again, a budget computer can give us very good performance. We can also then specify the template files. seqman engine can align to a wide variety of different template files. What we like to do is align to human genome template packages when we're working with human genomes. I could align to a FASTA file that's unannotated. If I just have a set of exons that I'm interested in and I don't really care about the GenBank annotations, that's perfectly suitable. Or I can pick a human genome template package. We create these packages for our customers and place them on our website. And what these packages are a combination of dbSNP. So in humans, I believe it's over 60 million variants in dbSNP. We have the Cosmic Cancer Database. The Genome Evolutionary Rate Profiling Database, GERP, is also packaged. And what we do is we bundle this together, and our customers can then download these packages for all the different model organisms and use them as a reference. And the advantage there is we have all the annotations with the GenBank files, plus all the dbSNP information. So that information can pass through our assembler and go into our output file, our BAM file. So we can do statistical SNP analysis on the fly as data is streaming through, and then have all the feature information to really facilitate our analysis. So we can see different model organisms here. There's multiple human packages. We have some ethnic-specific packages. We have different builds of the human genome. If your organism isn't present here, you can contact DNAStar, and we can look into building that package for you as well. So we specify what we want to align against. Then we can pick our read technology. So I'll take Illumina long reads. Now these days, most Illumina data is long over 50 base pairs, so that isn't too much of a problem. And then we can add our data. And so we can add both paired and unpaired data. I'm just going to add some unpaired data here. And I will. And what I'm doing here is I'm jumping up to, this is how it's optimal oftentimes to save and handle data. These are some Illumina FASTQ files. I keep them on a storage drive up on my network. And rather than keeping them on my local C drive, I'm just going to load a couple of FASTQ files here just to show you how we can set this up. So here's 3 FASTQ files, and these are human exome data sets. And I like to keep my data separate on one drive, and in this case it's up on my network. And I have a temporary drive to handle the temporary files, and then of course my operating system runs on the C drive. And in that configuration, I can get the best possible performance out of the assembler. Now I have some options here in terms of how to handle these different sets. I have 3 different individuals here. I could load multiple files per individual or load the whole folder in. If this is multiple individuals, I can select multiple sample data. And when I do that, I can assign a group name then to each one of my sets. Our software will automatically look to the FASTQ file name or the folder name and assign a group name, but I could name this something as well and just assign a group name. And what will happen then is when we run the assembly, these 3 samples will align to the reference sequence together. And for many different projects, it really facilitates the analysis to have all the projects together aligned to a reference. We can compare them directly. We can see which samples have SNPs, which do not, and the visualization is excellent. We also have the option to run multiple projects, but then save them out as separate projects. So I could have 100 different exomes here. And if I select Run as a separate project, it will create 100 separate exome assemblies, each for each individual. Now we have additional software called ArrayStar that can analyze 100 human exomes. So if I have 100,000 SNPs in each one, and I want to find out which SNPs are common or unique, I can run them as separate projects, load them all into ArrayStar, and find the SNPs of interest in 100 exomes, something that's very large scale. So it gives us a lot of flexibility as to how we can set the project up. I could also load paired data. When you load paired data, it'll ask you for the insert size, and so that's, again, another option for you. The assembly options, we generally will stick with default options here. We've run thousands of Illumina data assemblies in our software. We know that these parameters work very well for the data. If there is one parameter I would change, or at least experiment with, it would be the minimum match percentage. And this is the percent that the read must match the template in order to align. And I can increase the stringency by increasing this number from the default of 93 up to 95, and I can make the assembly a little bit more stringent. In some cases, if your reference genome maybe is a different strain and you expect there to be a large number of differences, I might lower this a little bit, maybe down as low as 85%. Again, it's a single parameter that has the most impact on the assembly. We also pick genome ploidy. Again, this is for the statistical SNP caller. We tell it what ploidy the genome is so that it can assign the right genotypes. There are advanced options that I won't go through in great detail here. Alignment options, more knobs and dials to adjust. SNP options where we can apply some baseline filtering to reduce the amount of sequencing noise that passes through to the SNP report. So again, we have full control over the assembly, but generally we just stick with default options. What's happening now is we're generating a script in the background. And for most users, we don't want to run software from a script, but there are some users that need high throughput or like to automate and have completely hands-off operation of the software. And the script file is just instruction for the assembler to run the assembly. And I can do things like copy and paste multiple scripts together and run 20 assemblies overnight or something like that. So it does give you added flexibility if you're comfortable running software from the command prompt. When I click Assemble, we get a log that launches here, and so we can watch the progress of our assembly. If there's a problem, if there's a data problem or a hardware problem, we'll see some error messages here. We can export the log and send that to Matt at DNAStar, send it to me, and I can help troubleshoot with some of my colleagues. I also like to keep my task manager open, and we look at XNG, and we can look at the CPU usage. And just to show you how the hardware is utilized, right now it's 98%. So I'm risking disruption in my webinar here a little bit, but the software is optimized to use every bit of hardware, at least initially while it's processing the majority of the data. So it's highly, highly optimized software. It's using a nice chunk of RAM, and it'll get you the fastest possible assembly on the hardware that you have. So I'm going to stop this assembly, and the output then can be opened in the seqman Pro module in the Lasergene suite. Of course, DNAStar has customers worldwide and for many years, so there are many users of the seqman Pro software. seqman Pro allows us to visualize the output from seqman Engine. So seqman Engine will create a bam.assembly file or a seqman file. We can open this in seqman Pro and take a look. I've got windows, a bunch of windows open here. I'll explain what they are. This first window is going to show a list of contigs. This is an assembly of 8 exomes to human chromosome 1. So we can see the length is 250 approximately megabases. And I can double click here and go right to an alignment view. The alignment view shows us lots of different information. This top sequence here with the twisty triangle is the reference sequence. And the reference sequence has annotations on it that I can reveal. And we can see gene features, CDS, mRNA. Below it, we have the aligned sequence data. And again, these are ethnic groups. And right now, it's fully expanded. So if I click the twisty triangles, I can collapse the assembly at that point. So again, this is a multiple sample assembly. It's been multiplexed in Engine. And I can collapse things down, and now I have what we call a pseudo-consensus, and it will show me the differences, if there are any, in the aligned data. So I can see at this point, I've got a reference sequence that's a C, and one of the samples has an ambiguous call, has a variant call. And it's hard to see it, I think, in a webinar, but this is actually a green C. That tells me that that's an annotated SNP, and it has been present in dbSNP. If I want to look at the aligned data for one sample, I click one of the twisty triangles. I can scroll down, and I can see my aligned sequence data. So I can see that I have a T variant at that location, which is pretty certainly a heterozygous SNP. Now, of course, something that's as large as human chromosome 1, it's not efficient just scrolling back and forth through the chromosome. So I usually will navigate using something like the SNP report. I can also navigate using a strategy view. So under our contig menu, strategy view, we have a view that is interactive with the alignment view. So if I click anywhere in my alignment view, it will bring me to that point in the strategy view, which is more akin to a genome browser type view. So it's panned out. I can see a number of things that are interesting. These red areas are areas that exceed my expected coverage. And so we get these little red plots. The depth of coverage here, and I'll just expand this a bit. You can see the depth of coverage over some of these areas exceeds or almost approaches 1,000x coverage. And so these peaks correspond to targeted exons in the exome analysis. The features I can hover over and get information from the GenBank annotation. I can also scroll, again, like a genome browser. It's interactive with the other views. I can launch a SNP report, click on a SNP, and it will advance to that SNP in all the views that are open at that time. Now, the SNP report also contains lots of information. It's also the most commonly used interface for doing work with the project. So the SNP report, the way it's structured is that there are filters at the top. Each row represents a different variant in one of the samples, and each column represents a different set of information. So depending on what you're interested in, you can apply different filters and organize the SNP report, and then use the SNP report to navigate. You can double click and go to those places in the assembly to verify SNPs and look at the actual assembled data. I can also verify and start to build a custom SNP database, again, depending on what it is that you're interested in doing. Many different things are possible with this report. So again, I can go and look at all those areas. The SNP report, just to show you some of the filters, we can differentiate between annotated. Those are the SNPs that are in dbSNP that have RS numbers. So here's a SNP that's annotated. I can go to my SNP menu, open dbSNP. It will use that RS number to look up the entry in dbSNP and show me all the information that there is about that particular variant. There's also minimum and maximum filters, depth filters, distance from coding feature filters. I can show all SNPs or SNPs that create coding changes, and I get a total number here of SNPs that occur. And then I won't go through all the columns, but we have differentiations between the different samples, the MID column, the reference position. We get a genotype call. The splice column is whether or not the SNP affects splicing of exons. We have the impact of the protein, the percent of the SNP. The P-naught ref, which is a probability that it's not the reference sequence. Our GERP scores are here, all the feature information. So again, it's a great report for doing SNP analysis. It's fully interactive with other parts of the software. We can save the SNP report out to an Excel sheet if needed. So it's really a top-notch interface for working with large projects, multiple sample projects. Now, if I have other questions, like, for example, that I posted earlier, how do I do SNP associations? I've got 8 exomes here, and if I want to answer questions like which SNPs are in common to all 8, which SNPs are unique to each of the 8, that's a much wider scale, broader scale type analysis. seqman allows us to look at specific areas, specific genes, but for those broader scale associations, we use our DNAStar software. And with that, I'm going to stop sharing for a moment and go back to the PowerPoint just for time's sake here. And so DNAStar is part of the DNAStar Genomics suite. It's a program that originally was designed for microarray analysis. And so it has the infrastructure to bring in large sets of data with thousands or millions of data points, and then apply bioinformatics filters to that data and find interesting gene sets from the data. And then also porting in things like annotations from different databases to learn more information about those gene sets. It quickly evolved into an RNA-seq analyzing program. And then as we developed our human genome assembler, we had a need to be able to compare millions of SNPs between different versions of the assembler. And it works so well internally that we decided to commercialize it, and it's been very popular with our customers ever since. So we can start a SNP project, and we can set up our experiment list then. We can import a seqman engine assembly directly into DNAStar. Again, if I have a large data set, in this case a couple hundred exomes, I can run them as separate projects in seqman engine, just like I showed you in the wizard. Collect them in a folder, load the whole folder, and DNAStar will bring in, and we've tested up to 200 human exomes. It will identify all the projects as different experiments. There are some quick buttons here that allow us to set the experiments up however we want to. We may have additional experimental attributes. In this case, we have some controls and a disease data set. I may have additional information like a set of individuals with high blood pressure or different phenotypes. DNAStar has all the infrastructure to set up your project that way. Once I have the project set up, really it becomes the challenge is the filter. I've got millions of SNPs potentially. I want to find the SNPs that are interesting, or I want to find the set of genes that are most impacted by different types of SNPs. So I can filter then through my SNPs at either the gene or the SNP level. And I can then apply specific criteria for those SNPs and create sets. In this case, we're looking for non-synonymous SNPs that result in nonsense or frame shifts. I can apply different stringencies for the SNPs and then start creating sets. And then we can start to compare those sets. So here's a set of control SNPs. And I can see how many genes do we have, in this case, 28 genes. So I can cross over from SNP to gene analysis. Once I've created interesting sets for my control group, my disease group, or different ethnic groups, I can do simple things like Venn diagrams. And you generate a Venn diagram. There are some quick links in the software where I can show gene tables of the intersection or the unique sets. So it's, again, very easy to use. So here's a gene table for this particular experiment. This MLL2 gene was of particular interest, and it drops out. It was in our intersection along with some other potentially interesting genes. The table is populated in this case with Go consortium terms. These are all links. These are Go ID terms. DNAStar has an expanded capacity to bring in database information to populate this table with information that may be interesting to you. You can bring in a VCF file, for instance, bring in, depending where the VCF file originates, you might have other database information like Polyfen or OMIM, or again, other annotation databases. So great, easy to use software, allows you to find areas of interest. So with that, that was kind of our run through of GWAS, or a multiple exome type analysis. I'd like to go through another project that's unique to our software. It's the automated bacterial genome closure, and we like to call this an easy solution for what really can be a difficult project. And I've worked with many, many customers who have sequenced microbial genomes, and you sequence them. The de novo assembly is fairly straightforward, but it's the gap closure, it's the finishing of the genome that can be very, very difficult, especially for people that are new to genome closure. And so DNAStar has many years expertise, back from when our scientists assembled E. coli, and had worked out some very nice workflows for closing genomes. But they were manual, and it required a lot of expertise to do the closure. And so we thought it would be a great idea to make this a lot easier, both for ourselves and for our customers, to automate some of the more difficult steps. And so anytime that you have a microbial genome and you want to assemble it, you really have some choices to make. And the choice might be, do I do a de novo assembly? Is this a strictly de novo approach? Do I take my MySeq data, get the longest reads possible, paired end, I assemble it, get some contigs, then I use the pair information to order them in the scaffolds, and then once I have scaffolds, I try to close the gaps between the contigs. If your organism is, or what you've sequenced is completely novel, that's the only option that you have. However, if there is a reference genome that is closely related, you can use that reference genome to guide most of the assembly. And depending on how close it's related, you may have anywhere from just a handful to a couple of hundred or more significant differences, structural variants, larger insertions and deletions. And of course, the more structural variations that you have, the more difficult it is to use that reference genome to guide the assembly. And so what we've developed is a way to use that reference genome to guide the assembly of 90% of the genome, but then automatically identify those areas where structural variants occur, and then de novo synthesizing those regions. And again, doing it in a hands-off way so that the end user doesn't have to do too much manual gap closure, which is the difficult step. So this is just a little table here showing with a data set of E. coli data that produces, in a de novo assembly, 300 plus contigs. Some of these are small contigs, so there's probably a little less than 100 that you actually piece together to get your genome length. But it's a significant amount of work with 300 contigs. When we align to a reference genome, we get a single contig and we identify 31 structural variants. And using an older software or a program that does not have automated closure, you would manually resolve these 31 regions. You would have to split the reference, fill in the areas manually. It's going to be a significant effort to close 31 gaps. In our workflow, it's 3. So we go from 31 to 3 unresolved structural variants using the reference, which saves, you can plan on a couple hours per gap for someone that's experienced. So it can be several days' time for someone that's new to this kind of workflow. So really what the goal is with this automated genome closure, and this is our strategy view in seqman, and this is really what we want to see. And what we're looking at here are green areas in our strategy view, nice deep coverage. Here's one contig, here's a green area here. In this blue area, this is an area that was automatically filled in using this process. And we use the pair data with our Illumina data, and we use the pairs that anchor in the unique area in the contig and the pairs that fall into this insertion element. And we generate a contig there and then merge them automatically. So, and you can see these are labeled in a different way, a different code, so we know what type of sequences they are. So that's what we're shooting for. We want to see that happen at all these structural variations. So the way that we set the project up, again, it's with seqman engine. This is 2 closely related K12, E. coli K12 strains. We pick our project type, genome assembly. We have our different output options or different assembly types. We pick reference guided assembly with gap closure. This is limited to microbial genomes at this point, so about 10 million reads. That, again, with longer MiSeq reads, it's more than enough reads to get, an excellent assembly. We pick our template file. GBK files that are annotated are best. We load in the sequence data. For this type of a workflow, you do need to have paired end data. So it can be paired end or mate pair. You need the pair data to fill in those insertion areas. That's how those areas are filled in. So we set up an insert size. Doesn't have to be exact. Close is good enough generally for what that actual distance is. Sometimes it's nice too to have different size pairs if you can get them. We just stick with default options. We change our ploidy to haploid, and we're ready to go. And so we get an assembly, and the first place that I look in my assembly, I'll open the project in seqman Pro, and I want to see how many structural variations there are between my sequence strain and my reference sequence. In some cases, we don't pick the right reference. We go back to engine, and maybe we do a metagenomic assembly to 100 possible genomes. Engine can do that very quickly and identify what the best reference is. But this is just a picture, a screenshot of our structural variation report, and it shows me the reference position, the type, whether it is a insertion or deletion, and we just see the count here in the upper left corner that we have 31 structural variations. And so that just gives us an idea of how close these genomes actually are. If I had 300 structural variations, I would reconsider my reference sequence, or I might say, I'm going to go look at my de novo assembly. Maybe that's the better route. So I get an idea. Here's my starting point. We have 31. And the way these structural variations are identified, an insertion has trim points to where the sequences are trimmed back to the insertion point. So our algorithms find these areas automatically. Again, I'm showing you this because manually doing this can be quite difficult. You have to find all these locations, split the reference. It's quite the process to do this manually. Deletions are obvious sometimes with depth of coverage where it drops off on the template. We also have reads that map to either side of the deletion. Our strategy view allows us to show just those split reads. And when we look at the final output assembly, so in this workflow we actually generate 3 assemblies. I'm showing you parts of 2, but this is the output that we want to see. And I just showed the report, and where you see all this merging, that's all good stuff happening. That means we have contigs that are going together automatically. And what we end up with is a scaffold, and the scaffold has in this case 4 contigs in it. That means there's 3. A scaffold is an order of these 4 contigs, and there's 3 gaps in between them, and they're unresolved gaps. So I've gone from 31, down to 3 gaps, and I know from this organism these are just some larger. These are some phage insertions in this particular genome. So I've just eliminated a large percentage of the manual effort, and this assembly takes about an hour of time. So really a great way to speed up some of the assembly and analysis of microbial genomes. So with that, I'd like to move to the last, we'll call it a workflow, and that's a de novo assembly, and that's going to be up on Illumina's BaseSpace. So BaseSpace has some great advantages for Illumina customers. For one, you can easily transfer your data from your sequencing instruments over to BaseSpace. So one of the big challenges is sometimes just moving data sets around can be one of the bigger challenges with next-gen data. And so Illumina has things set up so that it's very easy to move the data over to BaseSpace, and then you have access to a number of applications there. And it's a growing list, and more people have been using these applications, and you can try them out and see how they work for you. Some are free, some you have to pay to use. Our current presence on BaseSpace is our seqman engine for de novo bacterial genome assembly. And this is a standard workflow in seqman engine. We looked at the interface a little bit, but we've essentially simplified the wizard, and really, because we know exactly what type of data people will be using, we can simplify that and make it easier for Illumina customers to use. And so I'm going to jump back here and go back to my desktop so we can look at BaseSpace. And hopefully all my tabs are still there. And so here is, so this is my account. So I created an account a few months ago, actually back in 2012. And I can kind of see what I've been up to lately. I have a couple of runs, a Bacillus cereus data set, a PHI-X data set, with a number of projects, and I also have a number of application results. There are different applications that are available on BaseSpace, and DNAStar's seqman engine is one of the applications. These links will bring you to our website, and I'm assuming the other websites for the other companies. There's a number of different data sets. There's some public data sets that are available, all different types of data. I also have my own projects, so we can add additional projects. And once you have data that's loaded and ready to work with, we can just click on one of the projects, and it'll bring this up. So I can bring up this particular project, which again is the Bacillus cereus project, and we have a number of different runs, BC 1, 2, 3, 4, all the way down. So I have a number of data sets that I can work with, and now I can just pick my application. So again, here's seqman engine. And so seqman engine then, again, with the cloud application, it's just very simplified. So I can pick my sample. I can give it a name. I can select my pair distance, usually to stay with 500. And then I can also estimate my genome size. So it's as simple as that. And then I just start my assembly. And we'll get a notice here that thank you, and then you'll get an email from DNAStar when the assembly is complete. De novo bacterial genome assemblies with Illumina data will take anywhere from about 15 minutes on the shorter side up to maybe an hour, depending on how much data you have. So you'll get a link from DNAStar to tell you when the project is ready. You can take a look at the project again back on BaseSpace. If I go to my dashboard, I have some app results that are over here in the right-hand corner. I can click on my link, and I have, again, a link for that run. And we can let that load up. And so the output from seqman engine when we do a de novo assembly is going to be a seqman project file. And a seqman project file will have the .sqd extension. And the .sqd extension then will, we can open that in seqman Pro. You can also download a BAM file. That BAM file can be opened in a BAM viewer as well. So it looks like, let's see here. I'll try that one more time. And I'll try a different project. It's not coming up for me here. Okay, so here are the output files. We have links for a report, and the report actually gives us lots of information in a de novo assembly. So we get the version of seqman engine that was used, the assembly time, and actually this was 12 minutes, so it was faster than what I had mentioned earlier. And the number of contigs, we have 50 contigs, the number of contigs to reach the genome length of 5,023. This is actually an excellent assembly, and it's something with MySeq data, with the longer reads and the high accuracy, you can really get good de novo assemblies. And so you see that it doesn't take many contigs to get to our genome length. The contig N50 is 253 kilobases. So if you're familiar with de novo assemblies, you know that that is an excellent, excellent benchmark for an assembler. And as we scroll down, we can see some more metrics, lengths of assembled sequences, the quality, and assembly parameters, and the scripting that was used to run the assembly. So the project report is really a great point just to see, you know, was my assembly successful? And then I can also go into the DNAStar folder. And again, here's the .std file that's zipped up. You can download this file. You can contact DNAStar, request a demo if you want to go and look at the project and work with the project, you know, for your analysis. So again, that's all. Thanks for your time and joining us today. I know I went through a lot of things fairly quickly. You can contact us for more questions. I'd be happy to answer all your questions. Also, you can set up webinars with me or my colleagues, and we can go through your workflows and answer any specific questions that you have on your project. So again, thanks, and I'll like to pass this back to Raymond.
Thanks, Matt. I see that there's a few questions that have come in on the webinar here for you. If DNAStar's default parameters don't work for my assembly, can I change assembly parameters easily? And how will I know which ones to change?
Yes. So all the parameters are all accessible in the seqman engine wizard. And I did show the one parameter, which was minimum match percentage, which really has the greatest impact on your assembly. And so that's the starting point. Depending on what the problem is with the assembly, we may go and change some other parameters like MER size or other things of that nature. But generally, what I recommend is go with the default parameters. If you want to improve the assembly, utilize our tech support. Give us a call, and we'll help you out and optimize that assembly for you.
Okay, another question. So why BaseSpace? What is the reason that DNAStar has embraced the cloud and specifically put their engine application on BaseSpace?
Well, we think that there's a number of obstacles that next-gen customers and Illumina customers face, and a lot of it's just logistics. It's how do I get access to my files? They're fairly large. They can be difficult just to transfer up and down on the network. With BaseSpace, the files, everything is set up perfectly so that you can move your data from the sequencing instrument right over to BaseSpace. So it's hands-off with the data. And then when you have a cloud app like seqman engine on BaseSpace, you can see the interface is very simplified. So you can run an assembly using someone else's hardware. It's not, you don't have to go and set up all the RAM that you need for the de novo assembly. You can use the hardware that's up on the cloud. So you have an advantage for hardware, and then also the downloading of the project. You can do some of the analysis up on the cloud and just download a completed project to complete your analysis. So it really streamlines the whole process for customers.
Great. There's another question about sharing. So how can they share their data or their results or the application with others?
So we have a number of options for licensing. So our desktop software provides networked licensing, and networked licensing allows licenses to float between computers. So you can share one or multiple licenses on an unlimited number of computers. That's usually within a lab, a department, maybe an institute. A cloud application also is a great way to share. If you have collaborators that are at different institutes, in some cases they may not have access to our software. A BaseSpace or a cloud app like the DNAStar Cloud can allow collaborations then between projects over great distances.
Well, that's great, Matt. Well, I want to thank you for your excellent presentation, and I want to thank everybody for attending. This has been the BaseSpace Push-Button Bioinformatics Webinar Series. The special topic today has been assembly and analyzing of Illumina data in DNAStar Lasergene Software. Again, thanks to Matt Kaiser for his presentation. I'm Raymond Tikotsky, your moderator, and I just lastly want to let you know that if you want to learn more about BaseSpace, you can either register as a user, where you see here, illumina.com/BaseSpace. You can also see BaseSpace in action on YouTube at youtube.com/users/Illumina Inc. Or if you're interested in becoming a developer on BaseSpace and to build some informatic applications for push-button bioinformatics, you can go to developers.basespace.com. Again, thanks everybody for attending, and we look forward to seeing you on the next webinar.